HapMap project. HapMap project, which is also called as Haplotype Map Project of Human Genome, it was initiated in 2002. The project was a 3-year effort to construct a map of the pattern of single nucleotide polymorphisms which are present across the entire human genome. Initially, the researchers, they hope that SNPs to be scanned will provide a shortcut for identifying the DNA regions which are associated with the common diseases of the humans. MAP may also be helpful in understanding how genetic variations contribute to responses of the environmental factors on the human genome. This is the main page of HapMap project. HapMap project actually was a multi-country effort to identify catalog of common human genetic variants. It helped to develop the better understanding and catalog linkage disequilibrium patterns across the genome in several populations. In case of HapMap project, there were 270 samples initially. 90 samples, they were from Nigeria. 90 samples, they were from European descendant. 45 samples were from Beijing, China, and 45 samples were from Tokyo, Japan. There were 3 different phases of HapMap project. In case of phase 1, one common SNP was genotyped after every 5,000 nucleotides. Approximately 1 million SNPs were typed. In case of second phase of HapMap project, approximately 3.5 million SNPs were typed. And in case of phase 3, 11 global ancestry groups have been assembled and 1.6 million SNPs were typed in a total of 1,184 individuals. So there were 3 different phases of HapMap project, phase 1, phase 2, and phase 3. If we see the summary of the HapMap project, approximately 4 million SNPs were genotyped on samples of African, East Asian, and European ancestry. All genotype data is available in publicly available databases. With the help of HapMap project, we are able to examine linkage disequilibrium pattern throughout the human genome. High density single nucleotide genotyping is available now, and with the help of HapMap, now SNPs have been validated. Frequency, we know about the frequency of the SNPs, about assay condition. We can correlate structure of alleles in the genome with the help of HapMap project. So these are the 1,447 CNVR from 270 samples of human genome that have been genotyped during the HapMap project. Here is the marking of structural variations which are present on different chromosomes of human genome. Here we can see in case of chromosome 1, these are the structural variations that have been marked by red and blue lines. With the help of HapMap project, it has been estimated that modern human arose in Africa and replaced the other human species across the globe. Initially, the human being arose here and it moved to other parts of the world. So HapMap also helped to identify that how there was the movement of the human species from Africa to other continents of the world. What is HapMap? HapMap is a haplotype map of human genome. HapMap project was actually an effort to construct map of pattern of single nucleotide polymorphisms which are present throughout the human genome.